So our second speaker is Akiko Iwasaki, who's a viral immunologist. And as many of you know, many cancers in humans are caused by viruses. And the question is, how do these viruses evade the immune response? And Akiko will give some clues.
Thank you very much, Dan. Let's see if this, it is quite dead.
Do you need the links down?
No, it's fine, thank you. So thank you all for coming to listen to the talk. Today I'd like to focus our study, as Lloyd, I'm not a cancer biologist myself, and I'm an immunologist who's interested in virus infections, as Dan mentioned, and we're particularly interested in the immune responses that happen in the female genital tract. So using mouse model of genital herpes, we've been studying what happens at the site of infection. Let's try to use this. So in the case of these three major sexually transmitted pathogens, HIV has been shown, especially in the monkey model, of SIV infection to enter the female genital tract through the transformation zone area. And HPV infection occurs in the basal layer of the keratinocytes that are also close to the transformation zone of the, so where the ectocervix and endocervix meet is called the transformation zone. And you can see that this region is actually particularly susceptible to a variety of different pathogens. HSV, on the other hand, has a mechanism of entry that allows infection from the superficial layer of the epithelial cells. And of course, it can invade into the deeper epithelial tissues. So we've been interested in understanding how these viruses are detected by the innate immune sensors and how we might use the information of how these viruses are surveyed by the immune system to be able to generate vaccines. And so we became interested in the HPV infection a few years ago. Actually, this was kind of enticed by us getting the pilot grant from the Yale Cancer Center, and thank you very much for that. We've been sort of studying, and this first part of the talk will really involve collaborative work with Dan DiMaio's group. And so we've been looking together at how the cervical cancer cells impact on the immune system, especially the dendritic cells. So it's obviously an important disease. It's the second most common cancer in women, and 250,000 women die each year worldwide, especially in the developing world. And so this cancer it's accounted for by previous infection with various high-risk HPV types. And this graph down here shows you that 95% or more of the cervical cancer has an origin in HPV infection. So, and it can be, the early stages can be detected by the Pap smear. And the cervical intraepithelial neoplasia then turns into invasive cancer upon undergoing EMT, as you've heard from Lloyd just now. And so the stage in which we can intervene as immunologists and virologists would be to really hone in on the early stages of the cervical cancer development. And so this is a normal ectocervix and also other mucosal surfaces that are squamous epithelial cells that are target of HPV infection. And with the advent of recent prophylactic vaccines against HPV, we can now target and eliminate a lot of the new acquisition of the HPV in a large population. So this is wonderful. But we still have millions of women who are currently infected with HPV who may eventually develop cervical cancer. And so the immunotargeting would be done the best at an early stage of the cancer transformation. And this low-grade stage squamous intraepithelial lesion can be detected by the Pap smear. And when it becomes high-grade and invasive, it would be, I believe it would be more difficult to treat these cancers with immunotherapy. However, it is important to note that in all stages of the cervical cancer development, you see that these red nuclei here, which indicates the expression of the early genes, including the oncogenes E6 and E7, and even all the way out to the stage of invasive cancer, we can use the E6 and E7 as viral target antigen to prime T cells that are specific, that can kill these tumors, hopefully when they're targeted to these sites. And so the difference between the prophylactic vaccine and the therapeutic vaccine is that the prophylactic vaccines normally work by generating neutralizing antibodies that would prevent new incoming virus from infecting the host, whereas those antibodies are not efficient in clearing existing infections. So for the latter, we really need to generate cytotoxic T cells and Th1 cells that can recognize and kill these existing HPV positive cells. So I'd like to kind of spend the next few minutes talking about how this particular lesion is kind of devoid of immunosurveillance by dendritic cells, which are the most potent antigen-presenting cells, and also talk about the fact that there are, this lesion is usually filled with FOXP3 positive T regulatory cells that suppress the effector function of cytotoxic T cells and other T helper cells. So we need to overcome these two barriers in order to develop an effective therapeutic vaccine. So just to go over some of the dendritic cell types that can be found within the ectocervix area. The Langerhans cells are the predominant dendritic cells that are within this squamous epithelial layer, and we can identify them based on CD1A expression as well as CD207, also known as Langerhans. And whereas other types of dendritic cells that are present in the submucosa do not share all the same markers. So we can identify the Langerhans cells in culture by these markers that I just mentioned. And it's known that these Langerhans cells, unlike other types of dendritic cells, arise from a local precursor, and they're upon infection and invasion, they can arise from these monocytes that are circulating throughout the body. So what's striking about the intraepithelial lesions is that they're really kind of devoid of Langerhans cells. So this is one of many studies that demonstrate this. We also see this in our hands, that if you look at the squamous intraepithelial lesion, just next to the normal exocervix area, you see that there's completely devoid of the Langerhans cells. There may be a couple of signals here. This is CD1A staining. So somehow the lesion excludes or kills Langerhans cells that arrive there. So what are the potential mechanisms of this Langerhans cell reduction within the cervical intraepithelial neoplasia? There have been several theories. First is the disruption of E-cadherin expression, and this has also been shown by several groups to suggested to account for the lack of Langerhans cells, because E-cadherin is used by the Langerhans cells to be retained within these epithelial cells. The other thought is that the chemokine that's responsible for recruiting the Langerhans cells, such as CCL20, the expression of chemokines is selectively also reduced in the CIN. But we also wanted to address whether Langerhans cell differentiation from the precursor, which is really the main way you would normally generate Langerhans cells within these epithelial cells, whether that might be also blocked by HPV. So the question we addressed was whether the tumor microenvironment affect the differentiation of Langerhans cell precursor in situ within the cervical cancer lesions. So in order to do this, we utilized a well-established culture system in which monocytes so we collected monocytes from healthy human peripheral blood and treated it in cocktail of GM-CSF and IL-4 to derive dendritic cells, or GM-CSF, IL-4, and TGF-beta to derive Langerhans cells for 7 days. And we either added nothing, or we added UV irradiated HPV positive cancer cells to this culture to ask whether the cancer cell has any effect on the monocyte differentiation to Langerhans cells. And the data were quite striking, that if you just focus on the first panel here, this is the Langerhans cell differentiation and dendritic cell differentiation in the absence of cancer cell. When we added HPV negative cell lines, We've done several different cell lines that are HPV negative. The cancer cells have some effect on Langerhans cell induction, so you go from 88% to 60%. But when we added HPV positive, either 16 or 18 positive cell lines, we see quite a marked reduction in the Langerhans cell frequency and number in the co-culture system. So something about the HPV positive cancer cells are affecting the monocyte differentiation. So what are the factors that could possibly be doing this? So at the stage that I'm talking about, these cancer cells are expressing mostly the viral oncogenes A6 and A7, and we wanted to know whether those are sufficient to block Langerhans cell differentiation from monocytes, and is it dependent on cell-to-cell contact? So to address the first question, we went about to do these co-culture system with HPV negative cervical cancer cell line called C33A, which Dan had kindly provided for us, which also ectopically expresses viral oncogenes E6, E7, or both together. And what we saw was that, again, so if you compare the C33A alone to the E6 ectopically expressed C33A, we go from 78% to 45% frequency in the Langerhans cells in the co-culture. And this is not seen in the case of E7 expression, and dual expression does not increase this effect from the single expression of E6. So these data suggested to us that ectopic expression of E6 is sufficient to block Langerhans cell differentiation from monocytes. Now, this is also, you might notice that the effect of ectopic expression of E6 alone does not recapitulate what you see with the HPV positive cancer cell lines, indicating that there are probably other factors in the actual cancer cells that block monocyte differentiation in addition to E6. But since we had a handle on this sufficiency of E6, we then went on to study the role of E6 in blocking monocyte differentiation to Langerhans cells. And also we tested whether cell-to-cell contact was required for this effect. So the comparison would be, this panel indicates monocytes on the lower compartment of a transwell system, and the HPV irradiated cells are in the upper compartment, so they're separated by a membrane. And in that case, we don't see any reduction in Langerhans cell frequency or number, whereas when we use no transwell, we see the reduction to 13% frequency when we have overexpression of E6 and E7. So cell-to-cell contact is required to mediate this effect. So how does the HPV6 actually block the Langerhans cell differentiation? And to this end, we looked at some of the key partners or targets of E6 in its transformation activity, namely the E6AP, which is an E3 ubiquitin ligase, which ubiquitinates the targets such as P53, PDZ containing proteins and others to degrade through proteasome degradation. And of course, if you degrade P53 from the cell, you would have accumulation of mutations and inhibition of apoptosis. And PDZ domain proteins are involved in blocking transformation of the cells. So we first wanted to examine whether the proteolytic proteolysis of target proteins in general is required to see the effect that we see with the E6 overexpression in blocking the monocyte differentiation into Langerhans cells. To this end, we treated the cancer cells with lactocystin, which is an inhibitor of the proteasome, and asked whether when you inhibit proteasomal activity in the cancer cell, do we now restore the Langerhans cell differentiation activity? And that's what we see, that if you compare the DMSO, which is just a carrier control, the lactocystin treated cancer cells still support some differentiation of Langerhans cell compared to the parent cell parent cell alone, and E7 has very little effect on this. So we think that proteasomal degradation of E6 targets are likely playing a role in inhibiting monocyte differentiation into Langerhans cells. So we next wanted to see which of these targets are involved in the monocyte differentiation block by the E6. So in collaboration with Peter Howley's lab at Harvard, we were able to obtain E6 mutants that have specific inability to associate with either E6AP, P53, or the PDZ domain proteins. And using such mutants, so this has been mapped out already by Peter and other labs to diminish association with these target proteins that I just mentioned. We use these specific mutants to ask which of these target proteins are inducing this inhibitory effect. And what we found using another cell line, which is a human keratin cell line that's not HPV positive, Hackett cells, when we do overexpression of either the wild type E6 or the mutant E6, this experiment revealed to us that only the wild type E6, but not the mutants that cannot bind either P53 or the E6AP or the PDZ domain containing proteins, are able to suppress the Langerhans cell differentiation. So this told us that all of these domains are somehow required for inhibiting monocyte to Langerhans cell differentiation. And so what are the other consequences of this E6 expressing cells on the monocytes that differentiate in these cultures? And so we wanted to see the functional consequence of having the E6 during the monocyte differentiation on the antigen presenting function of the Langerhans cells. So what we did was, this is work done by Norihumi Ijima, a postdoc in the lab, who cultured, again, the monocytes with these HPV negative cells that are ectopically expressing E6 or the mutant E6 for 7 days. And then he isolated the monocytes and co-culture with allogeneic T cells for 5 days. And these T cells were pulsed with CFSE, so you can measure division of cells. And we also labeled with FOXP3, which is a transcription factor, master regulator of regulatory T cell differentiation. So using this co-culture system, he was able to show that the wild type E6 overexpressing cells are able to turn the Langerhans cells into a myeloid suppressor-like phenotype, in which these cells are now capable of inducing T cell proliferation and upregulation of Treg phenotype, the FOXP3 expression. And in this case, the P53 binding mutant was also capable of doing this, whereas these other mutants basically had a baseline activity. So the Langerhans differentiation in the presence of E6 expressing cells turn them into a suppressor-like cell that induces Tregs instead of effector T cells. So to summarize this part of the talk, we basically found that the high-risk HPV E6, but not E7, phenotypically blocks Langerhans cell differentiation from monocytes, and that all these domains, P53 binding, A6AP binding, and PDZ domains containing motifs are required for this effect, and the proteolysis of these targets are likely also important in mediating this effect. HPV E6 skews the Langerhans cell differentiation into a suppressor-like phenotype, which are capable of inducing Tregs. So implication of this study is that the future efforts to develop therapeutic vaccines for cervical cancer may need to overcome this blockade of monocyte differentiating into antigen-presenting cell in situ. And so there has to be a way of overcoming this and mechanism by which this HPV-positive cells do block the dendritic cell function within the lesion is going to be key in trying to prevent this from happening. Because this is not only important for the priming of the immune response to these antigens, but also for restimulating T cells that eventually get there. And so that's the other hurdle that I mentioned, is that perhaps a huge hurdle in the therapeutic vaccine strategy against cervical cancer is the paucity of HPV-specific effector T cell within the lesion. And this is common for many of these female reproductive tract tissues. Even normal tissues are not conducive to migration of effector T cells. And so the question is, how can we mobilize T cells to the site of these restricted sites, such as solid tumor, or in the case of female genital tract, the vaginal and cervical tissues. And so I'd like to spend the next few minutes talking about this new vaccine strategy that we've kind of came up with in order to overcome this restriction of effector T cell migration into certain tissues. And I believe this can be applied to any sites that are restricted for migration. So it's been known for a long time that memory T cells don't circulate equally throughout the body. There are sites of common entry, such as the lung parenchyma, lymph nodes, spleen, liver, bone marrow. These sites are surveyed by memory cells equally, have equal access without any inductive signal. Whereas other sites, such as the brain and the lung airways and peritoneal cavity, lamina propria of the intestine, and the genital tract are quite restricted in terms of memory T cell migration. And we've shown previously that the CD4 T help is required for CD8 T cell entry into the genital tract. And others have shown that resident T cell population in the skin, for example, are restricted from migration. They sort of serve as its own pool of effector T cells. And skin resident memory T cells also provide more superior immunity against HSV infection compared to circulating memory cells. And we also have exact same evidence for genital tissues that local memory cells provide a much more robust protection against virus infection in general. So then we wanted to overcome this restriction by the migration into these specific tissues by coming up with a prime and post strategy, which is a two-hit vaccine strategy. The first signal is just the regular conventional vaccine to prime T cells by giving antigen and adjuvant. But the second signal is to pull the T cells into the site of potential exposure, in this case the genital tract, using chemokines that we know that the T cells use to enter tissue. And so this is a very simple idea, but we wanted to explore this for protection against genital herpes. So we've been using the model of genital herpes to address these issues for many years. And so we use this sort of prime and post strategy to prime first with the TK minus. This is an attenuated virus subcutaneously given to mice, which induces circulating memory cells, but not tissue resident cells. We also had a control mice in which the same virus was given intravaginally, and this establishes local resident memory cells, and this is the golden standard for HSV vaccine. And then we come in later, 5 days later, with topical application of chemokines, CXCL9 and 10, which are ligands for CXCR3, which is expressed by the memory cells, or vehicle control. And then days later, so up to 90 days later, we look in the tissue for recruited T cells. So we are marking the specific T cells using TCR transgenic CD8 T cells that are specific to HSV. So what we see is that even though these priming strategy generated an equal number of effector T cells throughout the body, within the vagina, the prime and pooled mice are the ones that were able to retain the memory cells, CD8 T cells, long term, compared to, this is the golden standard intravaginal infection, as I mentioned, of course retains very well. But if you compare this to the just regular old vaccine in which you don't apply the pull signal, they really go back down to the naive mice level. And CD4 T cells are not retained very well using this technology. So basically, we asked whether this technique can provide protection against lethal genital herpes challenge. And we were very happy to see that the prime and pull provides this very robust protection against disease, as well as weight loss and survival. So essentially 100% of the mice that received the prime and pull were protected compared to just prime alone, which are these black circles. And of course the naive mice die much quicker because they just don't have any memory cells. So this provided us with a tool to be able to go after treatment, or even this case prevention, but even for treatment of diseases or infectious diseases or tumor, which are usually inaccessible by effector T cells. So a single chemokine treatment given in just saline, intravaginally applied, can recruit effector T cells, and effector CD8 T cells are retained for long term by mechanism we don't understand. It's just a single chemokine pull and they just retained. And disease from HSV infection is prevented by this method. So the future question is, can we apply this technology for example, cervical intraepithelial neoplasia, in which the effector T cells are very rare because they just don't circulate there, and many other situations in which solid tumors are devoid of effector T cells. So with this, I'd like to acknowledge the people who were involved in this work. So the first part of the talk with the HPV was really done in collaboration with Dan's lab and Edward Goodwin. And Norihime Ijima is a postdoc in the lab who did majority of the work, the data that I presented here. And the second part of the prime and pull was done by Hannah Shin. And Peter Howley and Elizabeth White at Harvard provided us with all the mutants that I described today. And again, thank you for Yale Cancer Center for giving us a couple of pilot grants to get us started on this. And I also received funding from NIID. Thank you.